Okay, just as we are talking, so what did you say? What did you say?
Histamine.
Okay, so that is when the DNA polymerase is synthesizing DNA. But remember, in this case, we are talking about RNA polymerase synthesizing RNA. So here, when RNA polymerase is synthesizing RNA, the pairing will continue like this until when RNA polymerase read adenine instead of thymine, because RNA do not have thymine. So RNA polymerase will add uracil. Is that clear? Is that clear? Class.
Yes, sir.
So instead of RNA polymerase, after reading adenine, to attach thymine, because it is synthesizing RNA, and in the RNA there is no thymine. Therefore, RNA polymerase will attach uridine or uracil instead of thymine. Likewise, if RNA polymerase read thymine, it will add adenine, because RNA polymerase, RNA contain adenine. Is that clear? Is that clear?
Yes, sir.
Okay. So this is how RNA polymerase synthesize RNA. So it depends, like we said, if it is RNA polymerase 1, it synthesize RNA, which is later on used for the synthesis of ribosomal RNA. If it is RNA polymerase 2, it synthesizes the messenger RNA, which later used for the synthesis of protein. And if it is RNA polymerase 3, it synthesizes the transfer RNA, which later on used also in the protein synthesis. We'll see how the protein synthesis happens. In protein synthesis, the process we call translation. Sorry, I put DNA translation here is a mistake. It's not supposed to be DNA translation. It's supposed to be
protein translation.
Translation.
My nose doing it.
So what happened in the case of translation? First of all, what is translation? Translation is the process through which an information in the RNA, basically the messenger RNA, is used to produce a protein. So this process of protein production involves the messenger RNA, the ribosomal RNA, the transfer RNA. But in all the cases, all of them function, produce, or are there for a different function. The information is coded in the messenger RNA. So the messenger RNA comes with the information on how the DNA will be, the protein will be synthesized. Remember, the protein is made up of what? It is made up of amino acid, right? It is the polymer of amino acid.
Mm-hmm.
The question is, which amino acid can be joined together through the peptide bond to give a particular protein? For example, insulin. You want to produce an insulin. Yes, any other protein is made up of amino acids. This amino acid, we made mention the 20 amino acid. All the proteins are made up of these same 20 amino acids. But how is this protein differ from another? Is the composition of the amino acids and the sequence of how they were arranged. Which one comes first, which one second, which one next, and how many is in one. How many amino acid is present in this protein could differ from the number of amino acid present in another protein. So that information is contained in messenger RNA. So messenger RNA comes with the information of the number of amino acids that this protein contains. It comes with the sequence, the arrangement, which amino acid comes first, which one comes second, which one comes last. All these informations are encoded in the messenger RNA. The work of transfer RNA there is just to read.
This
like the function of the transfer RNA there is to, first of all, the function of the transfer RNA is to bring the amino acids that need to be added. For example, we know that for messenger RNA, we have the nucleotide sequence. And based on what we call amino acid codons, this is something you need to know, that each 3 nucleotide code for 1 amino acid. Each 3 nucleotide code for 1 amino acid. So you have the ribosomal RNA, the messenger RNA will come and align itself to ribosomal RNA. So in the ribosomal RNA, there is position for messenger RNA, the transfer RNA. So you can have, when you look at this structure, let me see my cursor. So when you look at this structure, you can see this is transfer RNA. This one is also transfer RNA. But this big one, like this, can you see my cursor? Are you seeing it? Class? Are you seeing my cursor?
Yes, sir.
So this part is the ribosomal RNA. You can see the ribosomal RNA is 2 subunits. There is small subunit, there is a bigger subunit. This one and this one form the ribosomal RNA. So in the ribosomal RNA, you have the the messenger RNA attached to itself. You can see the messenger RNA here. The messenger RNA with the codons here, with the nucleotide codons. And here is the transfer RNA. You can see 3 transfer RNA here, 3 of them. The new one coming, the one that contains the peptide, on the other one going. So what happened is, I'm just trying to explain how the translation takes place, the protein synthesis takes place, even though this is not basically what you're supposed to go into the nitty-gritty of it. The most important thing you need to know is that the protein synthesis involves the information from messenger RNA, and the ribosomal RNA and the transfer RNA play an important role in the synthesis of protein. Is that clear? Class?
Yes, sir. Yes, sir, it's clear.
So let me just
Yes.
let me just make it a simpler way so that I don't have to go through the nitty-gritty. Maybe in the next, if there is another course that will talk about the nitty-gritty of all this process, Then we'll talk about that if eventually I happen to be the person to take that. So what happened in the protein synthesis is that the information for the synthesis of the protein is brought by the messenger RNA. So the messenger RNA contains the information as to what and what amino acids need to be joined together to form the protein. How many of these amino acids? So the information comes from messenger RNA. The transfer RNA help in bringing the amino acids and then the formation of the peptide bond, while the ribosomal RNA give them the room for the synthesis. So the ribosomal RNA will now accommodate the messenger RNA, the transfer RNA, so that they will check which amino acids are needed. Then the transfer RNA will bring the amino acids, and then the amino acid will be joined through peptide bond. Amino acid so it will read and bring the amino acid, attach, go and bring another amino acid and attach like this. As they are reading the sequence, they are bringing the amino acid, they are adding it until when you have a long peptide or a protein is formed. So then you have your own protein synthesized. So briefly talking about naturation and denaturation of DNA. Because we said that the two strands in DNA are held by the hydrogen bond, and this hydrogen bond can actually be disrupted, can be broken down either by the temperature or by pH, both the acidic and the alkaline pH, or even the salt concentration. So once you have, what is denaturation? Once you have this DNA losing its own helical structure, separating the two strands to form the single strand. This is what we call DNA denaturation. And the temperature at which half of the DNA helical structure is lost, the temperature at which the half helical structure of DNA is lost is called the melting temperature. Class, are we together? What is melting temperature?
No, sir, please come again. I don't understand, sir.
Melting temperature, the temperature at which the half of this DNA will be separated to form the double strand is called the melting temperature. Remember, we said the DNA strand, the two DNA strands formed a double strand through hydrogen bond. And this hydrogen bond can be broken, can be disrupted, so that the double strand will also go back to the single strand. The temperature can break this hydrogen bond. Another thing we also say pH, both alkaline and the acidic pH can break these hydrogen bonds. The salt concentration can also break it. And we said melting temperature is the temperature at which half of the DNA will lose its own helical structure, leading to formation of the DNA single strand. Now, talking about the temperature, remember we said in the DNA, adenine binds to thymine through two hydrogen bonds, while guanine binds to cytosine through 3 hydrogen bonds.
Okay.
Now, between the adenine and thymine forming the 2 hydrogen bond and guanine and cytosine forming the 3 hydrogen bond, which bond is stronger? Is it the bond between adenine and thymine or the bond between guanine and cytosine?
Guanine and cytosine, sir.
Good. The bond between guanine and cytosine will be stronger. Therefore, if you have a DNA that contains high content of guanine and cytosine, meaning that that DNA need a higher temperature for it to be denatured. You have a higher temperature for that triple bond to be broken down. Is that clear? So any DNA with a high guanine and cytosine content will need a higher temperature for that DNA to be denatured. Is that clear?
Okay, sir. Yes, sir.
So the process by which this separated DNA can come back to form double strand is called renaturation. So breaking the strand to form a single strand, the double strand to form a single strand is called denaturation. Coming back together is called renaturation. Is that clear?
Yes, sir.
So I think this is the end of this class. And I don't know. I wanted to also, like I said last week, that I would try and join the two classes. So I think what I will do is basically it look like you guys are very tired. So I will stop this now, but I think I will record the other class and still share it with you. So for those of you that still can participate for the next class, I will make the recording for the next class. And in the recording, I will also talk about the component of the assignment in study session 4 and study session 5. Okay?
Okay, sir.
For those of us that can still follow me, I will end the recording of this one and then start the next one.
Now, sir?
Yes, now.
Okay, sir.
You can